My name is Yue Zhao, and I'm a PhD candidate studying now at the Francis Crick Institute in the UK. First of all, I want to thank the International Kidney Cancer Association for providing this opportunity for us to share our study, which is an integrated study of spatial dynamics and genomic alterations in renal cell carcinoma evolution from the renal TRACERx cohort. I have no conflict of interests to declare. So background, previous TRACERx studies which used multi-regional sequencing data to study intratumor heterogeneity and tumor evolution have found genetic diversity and chromosomal complexity in clear cell renal cell carcinoma. And in this study, we want to take one step further to extract the spatial information coming from the photos taken after surgery and integrate this spatial information with genomic data to study tumor evolution. And so what we did was to use a digital pipeline to extract the spatial positional information from the photos taken after surgery, which extracts the X and Y coordinates of each biopsy region and the boundary between tumor tissue and normal tissue. And then using that information, we reconstructed tumor maps, digital tumor maps for each tumor that we extracted, and use the information to do downstream analyses. So first of all, we divided those biopsy regions into two different groups using based on their based on the distance between each biopsy region to its relative boundary. And we saw a higher proliferation rate marked by Ki67, a higher necrotic rate, and higher burden of somatic copy number in the tumor center compared with tumor margin. And then we validated that experimental result using computational modeling in terms of necrosis. And the simulation model also suggested that there is a higher necrotic rate in the tumor center. And also we found that more pathological Fuhrman grade 4 regions and more subclones that metastasize were found in the tumor center. So taken together, our data suggests that central regions of clear cell renal cell carcinoma have more aggressive behaviors, which could be due to the harsh environment which cells in the tumor center face, such as hypoxia caused by inadequate blood supply and necrosis associated with ischemia, either as a chronic process resulting in gradual tissue damage or as an acute event such as infarction related to arterial or venous thrombosis, which will in turn shape the nature of tumor evolution. And finally, we calculated the pairwise genomic distance between each two biopsy regions and pairwise spatial distance between each two biopsy regions. And we saw a positive correlation between those two kind of distances. And that suggested that the further the two biopsy regions were apart, the more different their genomic backgrounds were, and which kind of reflect our intuition. And finally, the directions of growth were inferred based on the phylogenetic trees and the tumor maps we generated, and two types of the dynamics modes of spatial growth were found in our cohort, namely spatially contiguous growth and the dispersal-driven growth. So in our cohort, most of the clones follow the contiguous growth pattern, where the same clone expands contiguously, or parent clones give rise to child clones and expand contiguously one by one. And there were two cases had clonal dispersal, where the dispersed clone like shown here in purple and here in green. They were surrounded by an uninterrupted block of genetically different subclones, which mapped to the different branch of the phylogenetic tree. And take this one, for example, the green clone here gives rise to the green clone here, and they were not the nearby clones to each other, and they crosses the wall formed by the blue clones, which came from the other branch of the phylogenetic tree. We think this is a very interesting phenomenon, and that might be of some clinical meaning or some genetic events, some specific genetic events that will drive this kind of phenomenon. We're still trying to investigate further on this topic to see whether there's a clinical meaning of it. And finally, we want to thank all the people who provided immense support to this work. And also thank you for your attention.